Okay, let's dive in. Think about how animals grow. I mean, it's pretty amazing.
Absolutely. You start with something tiny and it gets, well, huge in comparison.
Yeah, like a kitten becoming a cat or a chick turning into a full-sized chicken. We're talking orders of magnitude increase in size.
Right. Massive growth during development.
But here's the really interesting part, I think. When you look at the adults, say adult cats, they're all remarkably similar in size.
There's a surprising uniformity.
So how do they do that? Especially when, even a tiny difference in how fast they grow early on could lead to really big size differences later. It seems like it should lead to more variation.
That's the puzzle, exactly. And it's what we're digging into today, looking at some fascinating research using these tiny worms, C. elegans.
Ah, the nematode worms, a classic model organism.
Indeed. And we're exploring how they achieve this body size uniformity at the whole organism level, which is a bit different from how we usually think about size control in just single cells, like bacteria.
Okay, so multicellular control. What's the main source we're looking at?
It's a research paper called Coupling of Growth Rate and Developmental Tempo Reduces Body Size Heterogeneity in C. elegans.
Got it. So for you, the learner, why tune into a deep dive about worm size?
Well, because understanding these really fundamental biological mechanisms, how growth is coordinated with development, it can offer some surprising insights.
Insights that might apply more broadly.
Potentially, yes. Insights into growth across different species, maybe even giving clues about growth-related issues. It's one of those aha moments, seeing how a seemingly simple worm uses complex strategies.
I like that. So our mission today is to unpack what this study found. How do these worms stay so consistent in size despite all the potential variation?
That's the plan. Let's break down their findings.
Okay, so first, let's really establish the challenge. You mentioned that growth during development is often exponential.
That's right. It means getting bigger at an ever-increasing rate, not just adding a fixed amount each day.
And the study highlights this, right? Saying even a small, like 10% difference in growth rate between two worms early on.
Could lead to really significant size differences later. They show this in one of their figures. It visually demonstrates how divergence can happen.
So the potential for variation is definitely there, which makes the final uniformity even more striking.
And biologically, it makes sense that there's pressure to hit the right size. Being too big or too small as an adult can affect survival, finding mates, reproducing.
Sure, natural selection would favor hitting that sweet spot.
Right. And while we know a lot about specific genes and pathways that promote growth or limit growth,
Like the brakes and accelerator pedals?
kind of, yeah, but the mechanisms that actually ensure uniformity across a population, that fine-tuning, have been less understood.
Okay, so was there a prevailing idea before this study? Any hypotheses?
Well, there was this idea of size thresholds.
Size thresholds. You mean like hitting a certain weight or size triggers the next developmental step?
Exactly. Like maybe insects only start metamorphosis when they reach a critical weight, or these C. elegans worms only molt to the next larval stage once they hit a specific size.
And is there evidence for that?
There is some, especially under specific conditions. The paper mentions that if you restrict the worm's diet, they do seem to reach a pretty consistent body volume before each larval transition.
Ah, okay, so under stress, maybe size becomes a key checkpoint?
Possibly. But the crucial point the authors make is that observing this under stress, like dietary restriction, doesn't automatically explain how uniformity is maintained during normal, healthy development when food isn't limited.
Right. It's like knowing how a car stops doesn't tell you everything about how it accelerates smoothly.
Good analogy. It may be part of the picture, but maybe not the whole story for normal conditions.
So if a simple size checkpoint isn't the full answer, where did the researchers look next?
They drew some inspiration from the single-celled world, bacteria, yeast, that sort of thing.
Okay. What have we learned from them about size control?
Well, in those systems, researchers identified a couple of key mechanisms, often called sizers and adders.
Sizers and adders. Can you break those down quickly?
Sure, a sizer is basically what it sounds like. The cell grows until it reaches a specific target size, and then it divides. It doesn't really matter how big it was when it started that cycle.
Okay. Hit the target size, then divide. Got it. What about the adder?
An adder is a bit different. Instead of aiming for a total size, the cell adds a relatively constant amount of volume during its cycle before dividing. So it's about the increment added, not the final size reached.
Ah, okay. Add a fixed amount, then divide.
Exactly. And both of these mechanisms help keep cell populations pretty uniform in size over many generations. They prevent cells from getting progressively bigger or smaller.
Makes sense for single cells. So the big question was,
Do these same ideas, sizers or adders, apply at the level of a whole multicellular animal like C. elegans, coordinating its overall growth and development?
right, does the whole worm act like a big sizer or a big adder as it goes through its larval stages?
That's what they set out to investigate.
So how do they actually measure this? You need some pretty detailed observations of individual worms growing.
They really did. The experimental setup was quite sophisticated. They used live imaging, watching hundreds of individual worms.
Hundreds. Wow.
Yes, and they tracked them right from hatching all the way to adulthood, continuously.
And you mentioned high frequency.
Extremely high. They captured images every 10 minutes.
Every 10 minutes, that's a lot of data.
It is. They kept the worms in these tiny microfluidic chambers, which helps with imaging, and used a strain that expresses green fluorescent protein, GFP.
Ah, so the worms glowed.
Essentially, yes, or parts of them did, making it much easier for computers to automatically detect their outline and measure them accurately.
Clever. And how did they get volume from 2D images?
They use computational methods to sort of digitally straighten the worm's image and then, assuming it's basically cylindrical, calculated the volume.
Okay, that makes sense. So what did they see happening over time? Did the worms just grow smoothly?
Not quite smoothly. They observed these distinct periods about 2 hours long where growth almost completely stopped.
Stopped. Why?
These were the lethargous periods, right before the worm molts its cuticle to move to the next larval stage.
Ah, the molting process.
Right. And then, after each lethargus, there was a sudden, rapid increase in volume, a growth spurt. These spurts clearly mark the transitions between the larval stages L1, L2, L3, and L4.
So periods of near zero growth, then boom, a jump in size.
Exactly. And using these patterns, they could precisely measure how long each larval stage lasted on average.
And overall, how much bigger did they get?
Over the course of development, from hatching to that final molt before adulthood, the average worm increased its volume about 40-fold.
40 times bigger. That's a huge increase.
It is roughly a 2 to 3-fold increase within each larval stage.
Okay, so they have these detailed measurements. What did they find about the rate of growth within those stages, between the molts? Was it just steady, linear growth?
No, and this is a really key finding. Within the larval stages, particularly L1 through L3, the growth was faster than linear. It was essentially exponential.
Exponential again.
Yeah.
So growing faster the bigger they got.
Exactly. Which, as we discussed, theoretically makes maintaining size uniformity even harder, right? Because small differences get amplified more dramatically with exponential growth compared to linear growth.
Right. It makes the puzzle even more puzzling.
They confirmed this by looking at the relative growth rate that's the growth rate normalized by the current size. It was nearly constant in L1 to L3, which is a hallmark of exponential growth.
Did it stay exponential all the way through?
It slowed down a bit in the final larval stage, L4. The relative growth rate started to decline, which they speculate might be linked to the overall developmental clock slowing down as the worm approaches adulthood.
Okay. And did bigger worms grow faster in absolute terms?
Yes, they did. They showed that individuals with a larger body volume at the start of a larval stage really had faster absolute growth rates.
Which fits with that exponential autocatalytic idea growth, fueling more growth, like a snowball effect.
Precisely. The bigger you are, the faster you add volume.
Okay, so we have faster than linear growth, which should increase variation, but we also know the final sizes are quite uniform. How did the actual measurements of variation stack up? Did they see big differences in growth rates between individuals?
They definitely saw differences in growth rates. There was clear heterogeneity. They calculated the coefficient of variation, or CV, for growth rate.
That's a measure of spread, like standard deviation relative to the average.
Exactly. And the CV for growth rate was around 6 to 12% across the different stages. So some worms were consistently growing quite a bit faster or slower than the average.
And was it consistent, like were fast growers always fast?
Mostly, yes. They found a positive correlation between growth rates in consecutive stages. If a worm grew fast in L2, it tended to grow fast in L3 too.
So no sign of slower worms catching up by suddenly growing faster later on?
Not really, no. It seems the inherent growth rate differences persisted.
Okay, but what about the variation in actual body size? If the fast growers stayed fast, shouldn't the size differences have ballooned?
That's what you'd expect. But, crucially, they found that the heterogeneity in body volume increased only moderately. The CV for volume went from about 6% at birth up to just under 10% by the L4 stage.
Only up to 10%. That doesn't sound like ballooning divergence.
It isn't. And importantly, it was significantly less variation than they predicted would happen if you just randomly shuffled the observed growth rates and larval stage durations among the worms.
Ah, so if growth rate and stage duration were independent, you'd expect much more size variation by the end.
Exactly. The fact that the actual variation was much lower strongly pointed towards some kind of active buffering mechanism. Something was keeping the size differences in check.
Okay, so this buffering mechanism, it's not a simple sizer or adder, at least after L1.
Yeah.
What did they propose instead? You mentioned a folder mechanism earlier.
That's right. They presented evidence showing that C. elegans doesn't really follow a strict sizer or adder rule for stages L2 to L4.
How did they show that?
Well, for a sizer, you'd expect worms entering a stage at different sizes to all exit at roughly the same size. And for an adder, you'd expect them all to add roughly the same absolute volume. Neither pattern really fit the data well for L2, L4.
Okay, so what did fit?
What they found was that the volume fold change within a larval stage, how many times the volume multiplied, was nearly constant regardless of the worm's volume when it entered that stage.
Fold change. So like every worm roughly doubled or tripled its volume during that stage.
Essentially, yes. The relative increase was consistent. They called this a folder mechanism.
Okay, a folder. But you said earlier that a pure folder mechanism could actually increase divergence, right? Because if you start bigger and double, you end up even further ahead in absolute terms.
Precisely. A pure folder mechanism where everyone just multiplies by the same factor, regardless of how long it takes, would still propagate and even amplify initial relative differences. So the folder alone couldn't explain the uniformity.
So there must be something else going on with the folder mechanism. What was the crucial extra piece?
This was the really elegant part of the discovery. They found a significant anti-correlation between growth rate and the duration of the larval stage.
Anti-correlation, meaning faster growth goes with shorter duration.
Exactly. Worms that grew faster went through the larval stage more quickly. Worms that grew slower took longer to complete the stage.
Ah, so the speed compensates for the time.
Yes. They termed this folder with growth rate compensation.
Okay, that makes intuitive sense. If you grow faster, you get less time, and if you grow slower, you get more time.
The end result is that you achieve a much more similar volume fold change despite having different growth rates. The time compensates for the rate.
So it's not about the slow worms catching up in absolute size, but about everyone achieving a similar relative expansion because the time allowed is adjusted.
That's the idea. The compensation allows individuals with different intrinsic growth speeds to execute a similar developmental program in terms of relative growth. They found that relative size differences tended to be maintained, but the compensation prevented them from getting wildly exaggerated.
That's a neat mechanism, very different from just hitting a size target.
It is. It suggests a coupling between the processes controlling growth rate and the processes controlling developmental timing.
Now, you keep mentioning after L1. What was special about that first larval stage, L1?
Right, the L1 stage stood out. The data showed that L1 did follow an adder mechanism more closely.
Ah, so L1 is an adder, but L2, L3, and L4 are compensated folders.
That's what the evidence suggests. Worms in L1 tended to add a relatively constant amount of volume rather than multiplying by a constant factor.
Why the difference? And is this L1 adder important?
It seems to be very important. The researchers ran simulations, and they found that both the L1 adder and the compensated folder mechanism in the later stages were necessary to reproduce the low level of size variation they actually observed in the worms.
So you need both. Neither one alone was enough.
According to their models, yes. The L1 adder might be crucial for initially buffering the size variation worms are born with, sort of setting them up within a narrower range before the compensated folder takes over.
Interesting, a two-part strategy. So how robust is this whole system, this coupling? Did they try messing with it, like using mutants?
And they did. They looked at several mutant strains known to affect growth or development, things like EAT2 mutants that have trouble feeding.
So they go slower.
Right. And others affecting signaling pathways like insulin signaling, RAGA1, or TGF-beta signaling, DBL1, LON1, or developmental timing genes like LIN14.
A whole collection of mutants affecting size and speed. What happened? Did the coupling break down?
Remarkably, for the most part, it didn't break down completely. While these mutations definitely changed the absolute growth rates and developmental rates, sometimes quite dramatically, the two rates generally remained positively correlated across the different genotypes. Faster growth still tended to go with faster development, and slower with slower, even if the absolute values changed.
So the underlying link seems pretty robust.
It appears so. However, deviations from perfect proportionality did affect the final adult size.
How so?
For example, the EAT2 mutants, the poor eaters, their growth rate was reduced more severely than their developmental rate was slowed down. So they didn't have enough time to grow as much relative to their developmental stage.
And they ended up smaller.
Exactly. They ended up smaller as adults. This showed that the ratio between growth and developmental rate is critical for determining final size.
Okay. But within a given mutant strain, did the compensation mechanism, the anti-correlation between growth rate and stage duration, still work?
Yes, largely it did. They showed that within most of these mutant populations, the faster-growing individuals still had shorter larval stages, and the slower ones had longer ones. The compensation was still active.
Even if the overall average size was different for the mutant.
Correct. And as a result, even though some mutants had more variable growth rates, the variation in their final body volume was still less than you'd expect by chance, suggesting the compensation was still buffering size heterogeneity. There was one exception. The EAT2 mutants seemed less buffered, perhaps because the feeding defect introduced other variability.
Fascinating. So the coupling is robust, but the precise ratio matters for final size. Where do they think this coupling comes from? Is it just two independent processes that happen to be linked?
They proposed it might be more fundamental than that. They suggested the coupling could be an intrinsic property of the underlying developmental oscillator that times the larval stages in C. elegans.
Developmental oscillator,
Right.
like an internal clock.
Exactly. Think of a molecular clock within the cells that cycles and triggers developmental events, like molting, at regular intervals.
And how does that relate to the L1 adder versus the later folders?
Well, another unique thing about L1 is that this developmental oscillator is actually paused or arrested for the first few hours after hatching. It only kicks in later in the L1 stage.
So the adder mechanism happens when the oscillator is off, and the folder mechanism happens when it's on?
That was their hypothesis.
Huh.
And they tested it cleverly. They used a fluorescent reporter that lights up when the oscillator is active. They could then divide the L1 stage into an early part, pre-oscillation, and a later part, post-oscillation. And they found that the early part of L1 behaved like an adder, while the later part, once the oscillator started, behaved more like a folder, similar to stages L2, L4.
Wow, okay. That strongly links the folder mechanism to the activity of this oscillator clock.
It really does. It suggests the way the worm grows might be fundamentally tied to how this clock ticks.
Could they mess with the clock directly to see if it affected size?
Yes, that was the next logical step. They needed to experimentally uncouple the oscillator speed from the growth rate.
How on earth do you do that?
They used another smart genetic tool. They engineered worms where they could control the degradation of a key protein involved in the oscillator called BLMP1. They could use a chemical, auxin, to make this protein disappear faster.
So adding auxin would speed up the clock.
That was the idea. And they found that light doses of auxin did indeed speed up the oscillations, measured by that fluorescent reporter, and it shortened the larval stage durations. Crucially, this happened without significantly changing the overall growth rate of the worms.
So they uncoupled them. They changed development speed without changing growth speed. What happened to the worm size?
They ended up smaller. Speeding up the clock without a corresponding increase in growth rate led to reduced final body volume.
That's really strong evidence. Tampering with the clock's timing, independently of growth, affects size.
Exactly. Higher auxin doses also disrupted the relationship and reduced volume, further supporting the link.
Did they have a theoretical explanation for why the oscillator speed might naturally lean to growth rate?
Yes, they proposed a potential mechanism using a mathematical model of a simple genetic oscillator.
Okay, math models. How did it work?
The model involved an activator and a repressor protein cycling. The key idea is that in any growing cell or organism, proteins get diluted simply because the volume is increasing.
Right, the same number of molecules in a bigger space means lower concentration, dilution by growth.
Precisely. And in their model, the frequency of the oscillator depended on how quickly the repressor protein was removed. If the repressor is stable and mainly removed by this dilution effect,
Then faster growth would mean faster dilution.
which would mean faster removal of the repressor, leading to faster oscillations.
So the growth rate itself could intrinsically set the tempo of the clock through dilution.
That's what the model suggested. Their simulations showed that the oscillator frequency could scale almost proportionally with the growth rate, providing a simple, built-in way to achieve the coupling they observed experimentally.
And the model also explained the uncoupling experiment.
Yes, the model predicted that if you artificially sped up the oscillator, like removing the repressor faster by other means, analogous to the oxen experiment, without changing the growth rate, you would indeed alter the final size achieved, consistent with their experimental results.
That ties it all together nicely. A potential molecular mechanism linking growth, dilution, and the developmental clock.
It provides a compelling hypothesis, at least.
Okay, wow. That's a lot to digest. Let's try to summarize the key takeaways for the learner.
Absolutely. I think the first big point is that C. elegans achieves its remarkable body size uniformity not by strictly hitting size targets after the first stage, like a sizer.
Right. Instead, it uses this folder with growth rate compensation.
Exactly. The volume multiplies by a similar factor each stage folder, but faster growers get less time and slower growers get more time compensation, keeping the fold change consistent.
And that crucial first stage, L1, acts like an adder, adding a fixed amount of volume.
And that L1 adder seems necessary, along with the later compensated folder, to buffer initial size differences and achieve the final uniformity.
Okay, and this whole system is coupling between how fast they grow and how fast they develop.
Appears to be deeply linked to their internal developmental clock, the oscillator, potentially through something as fundamental as protein dilution by growth.
And messing with that link, like speeding up the clock without speeding up growth, directly impacts their final size.
Precisely. It shows how intertwined these processes are.
So it really paints a picture of multicellular size control that's quite different, more dynamic perhaps, than the simpler size ratter rules we see in single cells.
I think so. It highlights this intricate coordination between growth and timing.
What's a particularly surprising or intriguing detail for you?
For me, it's the idea that the rate of growth itself might be a fundamental input controlling the timing of development. It echoes ideas about growth laws seen in bacteria, suggesting maybe there are some very deep, conserved principles connecting growth and cellular processes across vast evolutionary distances.
Growth rate setting the pace of the clock. That is pretty mind-bending. Okay, so here's a final thought for you, the learner, to ponder.
If this kind of growth rate dependent developmental timing is happening in worms, could similar mechanisms be operating in other multicellular animals? Maybe even in humans?
What would the implications be? Could disruptions in such a coupling play a role in developmental disorders, or maybe growth abnormalities, or even aspects of aging?
It opens up interesting questions, doesn't it, about how robust development is achieved and what happens when the coordination between growth and timing goes wrong.
It really does. It just goes to show how studying a simple organism like C. elegans can reveal such profound and potentially widespread biological strategies.
Absolutely. There's always more to learn from them.
Well, this has been a fascinating deep dive. Thanks for unpacking that research with us.
My pleasure. It was a great study to discuss.
And thank you, the learner, for joining us. Until next time.